Hello, the title of my presentation is Screening for Amino Acid Content in Peanut using Raman Spectroscopy, presented by John Cason with Bill McCutcheon, Nicole Cherry, Joshua Yuan, Dimitri Kurowski, Mark Burrow, and Charles Simpson. Peanut is recognized as a highly nutritious food source. It is high in protein and oleic acid content, which are both traits associated with a balanced diet and increased heart health. Superfood status is given to a food which contains all the necessary recommended daily allowances of the amino acids that are necessary to serve as building blocks for the body's nutritional needs. Peanut is lacking in two of these essential amino acids. Lysine and methionine are produced at low levels, but below the amount that is needed for the nutritional requirements of the average human. The purpose of this poster is to describe the Texas A&M AgriLife breeding program's research into increasing specific nutritional traits in cultivated peanut. To achieve these goals, we will conduct our research in several phases with objectives in each phase. Objective 1 in phase 1 is to couple lab-based proteomic techniques with Raman spectroscopy that will eventually allow us to screen our wild species collection. We will quantify essential amino acid content with these lab techniques such as LC-MS, and then use statistical analysis to identify methionine and lysine signatures using our Raman spectrometer. Once we are confident in our results, we will scan our wild species collection with the Raman spectrometer for elevated levels of the essential amino acids in question, and then verify this with the lab techniques mentioned before. In phase 2, we will identify genetic mechanisms that are associated with these elevated levels, and then begin the introgression program using traditional gene integration, marker-assisted selection, Raman Based Selection, and possibly even gene editing to move these genes into the cultivated peanut. And then finally, in phase 3, we will begin field testing for agronomic traits in multi-location replicated testing. Initial results indicate that Raman spectroscopy can be used to identify peaks that are associated with nutritional traits such as protein, carbohydrate, and oil content. In addition, we have used partial least squares discriminant analysis to distinguish between high oleic and low oleic cultivars with accuracies approaching 90%. We see great promise for this type of data collection. Raman spectroscopy is uniquely suited for use in high-value germplasm like that in our wild species collection. It is non-destructive and non-invasive, and it uses a laser light to excite the chemical bonds between the atoms, and then this vibrational signature creates a chemical fingerprint that can be associated with traits for phenotypic evaluation. Furthermore, we feel like that peanuts with all essential amino acids at levels that are needed for human nutrition will allow peanut to be used as a complete protein in the fight against world hunger. And then finally, the ability to make quick scans of large amounts of germplasm will allow breeders to develop large libraries for traits and germplasm that could then be used for Raman-assisted breeding. We are very excited about this project and see great promise in the results that we are obtaining at this point. Thank you very much, and we would like to thank the Texas A&M AgriLife Research for their support of this research, as well as the Texas Peanut Producer Board and the National Peanut Board. Thank you.